amazing platform of Infinity Learn by Sri Chaitanya. My name is Kiran Chaudhary and I am your biology faculty at Infinity Learn. Students, in this session we will discuss about translation process, one of the most important, conceptual, and with respect to NEET, like you can expect question in your coming NEET exam, right? Very important topic. So let us start and let's see how translations takes place inside cell. Before starting, I would like to tell you students, do visit to our website infinitylearn.com and check out the amazing courses and products that we offer there, and you can use them at straight 40% off by applying the code Kiran40. So do consider visiting to our website infinitylearn.com. See students, so what we need to discuss first is the term translation itself. So what does this translation means? I think this a little, so let us see what is the term translation means. Students, translation is a process where amino acids will be bonded to each other with the help of a peptide bond, and as a result there will be formation of polypeptide chain. So I repeat myself students, two things we need to understand before I start explaining you how translation will take place inside cell. So listen this very carefully. What is a translation process? During translation process, polypeptide chain will be produced. What is a polypeptide chain? A chain in which, a polymer in which amino acids are present and bonded to each other with the help of peptide bond. Second thing, in what manner, or I can say which amino acid and in what sequence or order they will be arranged in a polypeptide chain, that will be decided by the codons that are present on mRNA. So the work of tRNA will be to read the codons that are present on mRNA and bring the respective amino acids on the tRNA so that they can get or they can form peptide bond among them, and finally there is synthesis of a polypeptide chain. So that's exactly what is mentioned over here on the slide in these two sentences. So please remember, what is translation? Polymerization of amino acid, and secondly, as I told you, order and sequence of amino acid is defined by the sequence of bases or codons present on mRNA. So for example, if this is a polypeptide, this is an example of polypeptide chain, a protein, and there are several, not even there are several, but there are several types of amino acid that are present in this polypeptide chain. So which amino acid and in which order or sequence they will come, decided by the codes present on mRNA. I hope the introduction about translation process is clear to all of you. Now let us see the next thing students. Let us talk in little detail about the peptide bond. See students, in order to produce a polypeptide chain, amino acids needs to bind with each other, right? So if there are 10 amino acids, for example, in a polypeptide chain, so that means there will be presence of 9 peptide bonds among these 10 amino acids. So let us throw a little light. Let me explain you in little detail about a peptide bond. So what is the function of peptide bond? It will help to bind to amino acid, and I am pretty sure with your 11th class biomolecules knowledge, you must be aware of what is an amino acid made up of, right? So amino acid has a N terminus and a C terminus, right? N terminus means presence of NH2 group and C terminus means presence of carboxyl group, that is COOH group, right? So this peptide bond formation between two amino acid takes place through interaction of N terminus of one amino acid and C terminus of another amino acid. So, peptide bond will be formed between N terminus of one amino acid and C terminus of another, right. So, by interacting these two functional groups between the two amino acid, there will be formation of peptide group or peptide bond, peptide bond. Now, that you can see on the slide as well. So, see students, this is an example of one amino acid or first amino acid, and this is an example of second amino acid. See this is the N terminus, this is the N terminus of this amino acid, and this is the C terminus of this amino acid. Now, see here, this is the N terminus of this second amino acid, and this is the C terminus of the second amino acid. So, peptide bond will be formed between the C terminus and the N terminus of the two respective amino acid, and as a result there will be formation of a peptide bond. See, this amino acid got bonded to this amino acid with the help of peptide bond here. I hope this much is clear to all of you. Let us see what I need to explain you next is in little detail about ribosomes. Very important, they are the cellular factory for proteins, right. Ribosomes actually provides a platform, a place over which proteins can be synthesized, and hence they are termed as protein factory, right. So, let us see or let us try to understand in little detail about what are ribosomes with respect to translation process. So, see here students, when we talk about ribosomes, 2-3 important terms that you need to understand. First, I have already explained you, they are the protein factory, right. Second is, they consist of rRNAs and 80 different types of proteins. Very, very important. If somebody asks you what is the composition of ribosomes, they are made up of rRNAs, right, the structural RNAs, right, and 80 approximately, this number can range, but approximately 80 different types of proteins. So, this is the rRNA, and approximately 80 different types of protein makes the biological composition of ribosomes. Next that you need to understand here is students, if the ribosome, see what is the function of ribosome? Help in protein synthesis. So, when this ribosome is not in active state or is in inactive state, that means this ribosome is not participating in translation process. So, listen this carefully, whenever the ribosomes are not participating in translation process, they will exist as two units, as two different units, and if they are participating in translation process in polymerizing the amino acid, these two subunits will be combined and form a single structure. So, when ribosomes are in inactive state, that means not participating in translation process, they will exist as two units. One is the smaller unit and another is known as larger unit. You can easily make out with this diagram. So, this one is the larger one and this one is the smaller one. So, large subunit, small subunit. So, what is this? So, they exist in two subunits in inactive state, means when not participating in translation process. So, my dear students, please and try to understand this last line is, hello Sunil, good evening. So, see Sunil, notice this. So, this is how ribosome will look like, the two units of ribosomes will look like when they are not participating in translation process, and this is how the two subunits of ribosomes will look like when they are participating in translation process. Let us try to understand some more important facts about ribosome, students, very, very important. Now, you know, there are two subunits of ribosomes, the larger one and the smaller one. The larger unit of ribosome has two binding sites, right? It has two binding sites. One is the P site and another is the A site. P site is peptidal site and A is amino acyl site. So, let us try to understand what are these sites, where they are present exactly and what is their function, right. So, larger subunit, remember larger subunit has two sites, two binding site. One is the P site known as peptidal site and A is the amino acyl site. So, see students, this one is the A site and this one is the P site, right. This one is the A site and this one is the P site. A is the amino acyl site. That means here amino acylated tRNA will enter. What are amino acylated tRNA? Let me write it for you. A site binds to amino acylated tRNAs. So, see my students, what is an amino acylated tRNA? A tRNA that is covalently bonded to an activated amino acid. I repeat myself, a tRNA that is covalently bonded to an activated amino acid forms amino acylated tRNA. I repeat myself, amino acylated tRNA is a complex, right. It is a complex in which there is a tRNA covalently bonded to its respective activated amino acid. So, A site is the binding site for such amino acylated tRNA. They are the one, they are going to bring amino acids from the cytoplasm, right. Next, this one is the P site. P site is the peptidal site. So, what will happen here students? Here peptidal tRNAs will bind. What? Here peptidal tRNAs bind. So, on this P site, who will bind? Peptidal tRNAs. What are peptidal tRNAs? Are the one on which peptide bond formation is taking place. So, see here students, if you see this on this P side, you can see continuously there is formation, there is polymerization of amino acid going on. So, there is a formation of peptide bonds over this P site of the larger subunit of ribosome, hence the name P site and A site. Also notice students, this P site and this A site are present in close association, they are present in proximity, they are close enough, right, and it is required. If they won't be close, then peptide bond will not be able to form, right. So, since P site and A site are close enough, hence there is formation of peptide bond, hence there is formation of peptide bond between the coming or subsequent amino acids, right. So, amino acids will continuously, subsequent amino acid will continuously come on A and peptide bond will continuously get formed on site P. So, that is how it works, okay. And from this P site, the tRNA will exit. Which tRNA? Who has already delivered its respective amino acid, it will take exit from this P site there, right, in order to get charged again with its respective amino acid and can again circle and do this activity all over again, right. Next students that you need to understand is ribosomes also have another function, that is ribosomes can also catalyze the peptide bond formation, right. Who can? Ribosome can catalyze. So, ribosome has the ability to catalyze the peptide bond formation, right. Although students, peptide bond formation is energetically favored process, right, but presence of a catalyst is going to make this peptide bond formation process far easy and faster as well. And hence here the ribosomes are going to act as a catalyst for peptide bond formation. And why ribosome or how ribosome? Do not forget, these ribosomes are made up of rRNAs, and you are familiar that rRNA, especially talking like rRNA, has this ability to act as a enzyme, example ribozymes, and in that also if you talk specifically in bacteria 23 rRNA, right, 23 as rRNA will act as a ribozyme. So that's how ribosomes can catalyze the process, peptide bond formation process, because the rRNAs present in them possess that catalytic nature. I hope students each and every point that is mentioned on the slide is clear to all of you. Next, let's talk about what is a translation unit. So see students, remember till now before this class when I was explaining you transcription, I explained you what is a transcription unit. So similarly when we are discussing translation process, the time has come to discuss to address this point, what is a translation unit. So a translation unit will be formed with the help of an mRNA. So mRNA having start codon, stop codon, and in between the codons for the respective amino acids. So mRNA flanked with a start codon and a stop codon constitutes or make up a translation unit. I hope very simple, clear. Chaliye, let us see the next one, and I know you must be clear with this, that a start codon, who acts as a start codon? In the genetic code part I have already explained you, right, that start codon AUG functions as a start codon. AUG has dual function, start codon and codes for methionine amino acid. Let us see the next portion students. There is another interesting discussion related to untranslated regions, in short UTRs given in your translation topic. So these untranslated units are actually the additional sequences, right, present on the mRNA. They are present on both the polar ends. They are present at 5' as well as they are present before 5' end on mRNA as well as after 3' end of the mRNA. Not to forget, they will not get translated during the translation process. That means the code or the nitrogen bases that are present in them do not code for any amino acid, but they are important for efficient translation process to take place, right. So what are UTR? Additional sequences on mRNA. They are not translated, present on both the ends before start and after stop codons, right. And very important, what's the function? For efficient translation process to take place, right. I hope this much is clear to all of you students. Now, my dear students, we have finally come to a point where I am going to discuss with you in detail about the translation process. That means from now for the coming few minutes I will explain you how exactly process of polymerization look inside a cell. So let's see, right, step by step how polypeptide chain inside a cell is synthesized. So there will be 5 steps, 5 major steps. I repeat myself students, the protein biosynthesis takes place in 5 steps or 5 stages. This is a complex process, right, one of the complex process in a biological system, that is protein synthesis, and heavily energy requirement or heavily energy dependent process this is, right. Please remember, very conceptual, very tough, very complicated. So that entire process of translation I am going to explain you with the help of these 5 stages, right. Although just the name of the stages is mentioned over there, on the next slide I will explain each and every stage in detail with the help of a proper diagram. So let me call out the names of the stages first, and then we will try to understand each and every stage in detail. The first one is activation of amino acid. Amino acids will be activated in presence of ATP. Second is initiation, right? Starting of the translation process, elongation, continuous polymerization of the amino acid. Termination, stop of the translation process when the required polypeptide chain is produced, and the remaining the ribosomes will be recycled so that they can again take part in the cycle of translation. And last, there will be folding and post translational changes in the protein that is synthesized after this process, right. So, now let us check out in detail what are these stages that actually helps in understanding protein synthesis, that is translation process. Okay, so here it goes, students. So, see here, I have to start with this point, but I want to explain you, this is a beautiful diagram to explain translation. So, with this image, I will explain you all the steps in translation process. See students, what is this? Please focus on this image. I hope it is visible to all of you. This is your tRNA. This is your tRNA. And this is, my dear students, your activated amino acid. Now, see students, listen carefully, very interesting this is. Amino acids will be, first of all, right, in the initial phase only, amino acids will be activated in presence of ATP, and such activated amino acid when binds covalently to their cognate or related tRNAs, that process is known as aminoacylation of tRNA, or that process is known as charging of tRNA. I repeat myself students for your benefit, okay. So, first of all, amino acids will be charged, amino acids will be activated in presence of ATP. Then this activated amino acid will covalently bind to the cognate or related tRNA. This process is known as, this process of covalently binding the activated amino acid to this cognate tRNA, right. This step or reaction is known as aminoacylation of tRNA or charging of tRNA. And please notice, this reaction is going to be catalyzed with the help of an enzyme known as aminoacyl synthetase enzyme. I repeat myself, aminoacyl synthetase enzyme. As a result, what you will get, a complex in which there is an activate amino acid and a cognate tRNA. So, see here students, this is the activated amino acid, this is the cognate tRNA. What is a cognate tRNA? The related one which needs to bind with this particular tRNA. Remember, for every amino acid there is a particular tRNA, cognate tRNA. This is a cognate tRNA. Alright. Now, please notice students, this reaction of covalently binding of a cognate tRNA with an activated amino acid will be catalyzed with the help of an enzyme. Enzyme is aminoacyl synthetase. So, I am writing here students, there is no proper space over here. So, I am writing here, I am drawing an aster here, and here I am mentioning what is the meaning of aster. What is the meaning of aster? Aster is the enzyme, aminoacyl synthetase enzyme. Okay students, so this reaction will be catalyzed in presence of aminoacyl synthetase enzyme, right. Now, this tRNA, remember students, we are now starting the process of translation, and if you remember my lecture, the last one, there I told you there is an initiator tRNA that is going to start the translation process, right. Since I am trying to start this process, I am trying to explain how this is how the translation process is starting up. So, this tRNA, if I say this tRNA is that particular tRNA that is going to initiate or start the translation process, start the polymerization of amino acid, hence this is known as initiator aminoacyl tRNA. It is initiator aminoacyl tRNA because it is going to initiate the translation, it is going to start the translation process. So, this is the initiator, aminoacyl tRNA. Next, this tRNA, this initiator aminoacyl tRNA will bind to this. What is this? This is the small unit of ribosome. So, see my lovely students, mRNA, this is the mRNA. This mRNA has start, oh I can't draw this aster here. So, this mRNA has start as well as stop codons, right my kids, right. So, this is the mRNA having start and stop codons. So, when this mRNA comes in contact to the small subunit of the ribosome, that is what your NCERT says, please notice this. This mRNA comes in contact with the smaller subunit of ribosome and also get binded to the initiating or initiator aminoacyl tRNA. Finally, this complex will be formed, right, where you can see my kids mRNA, right, smaller subunit of ribosome and aminoacyl tRNA, that initiator one. Later on, the larger subunit, this is what my kids, this is the larger subunit of ribosome. So, finally, with this complex, there will be addition of the larger subunit of ribosome and hence finally there will be a formation of a complex, this is a complex, this is the initiator complex or initiation complex, this is the initiation complex. See my students, finally there will be formation of an initiation complex where you can see smaller subunit of the ribosome, you can see mRNA, initiator, aminoacyl tRNA and the larger subunit of the ribosome. Now togetherly this complex will properly initiate or start the translation process, right. So, till here I have mentioned in points on this slide. Let me read out for you, but I have already explained everything when I was explaining you this much part. So, charging of tRNA or aminoacylation of tRNA will happen. Secondly, mRNA binds to the smaller subunit of the ribosome and to the initiating aminoacyl tRNA. Finally, large ribosomal subunit then binds to form an initiation complex. Now, see my students, what is going to happen next? What is going to happen next is this initiator tRNA. See, this initiator aminoacyl tRNA, this initiator aminoacyl tRNA will do what? It will bring it is going to bind at what site? It is going to bind to this mRNA with the help of the complementary base sequence that is it will bind to the initiator or the starting codon or the start codon, that is AUG, right students. So, this initiator or initiating aminoacyl tRNA with the help of complementary base pairing will bind to the start codon, that is AUG, right. Now, also note students, very important is now this whole process of initiating this translation process, this binding of the tRNA, aminoacyl tRNA to the start codon, and starting this translation process requires energy. Yes. So, GTP energy will be utilized, as well as there will be requirement of initiation factors, right students. So, this step I am writing here, this step, which step? Initiating amino acyl tRNA base pairs with AUG and signals the starting of the polypeptide formation chain. So, this initiation or starting of translation process, this step requires GTP and initiation factors. Right. Next, what will happen students? Next, elongation will take place, right. So, subsequently amino acids, one after the other, amino acids will go on coming to the A side, moving towards. So, amino acids subsequently will go on coming with the help of their cognate tRNA, will go on coming to the A side, will move to the P side, get the peptide bond made, and this is how the cycle will go on until and unless the entire mRNA is translated, right. See, next point is very important, amino acid, in what sequence and which amino acid, in what sequence and order amino acid will come during the formation of polypeptide chain, that is dictated by the DNA, and it is executed by or represented by the mRNA. Do not forget, this is a very beautiful line, dictated by DNA and represented by mRNA, very, very beautiful line. So, this elongation will take place. Next, see students, this is how elongation will go on and go on continuing, right. And not to forget students, even this step requires hydrolysis or involvement of GTP energy, hydrolysis of GTP, right. So, initiation also requires GTP, elongation also requires GTP, not to forget, right students. चलिए देखिएगा यहाँ पर क्या हो रहा है. See, listen here, what is happening? Another, what is this? Another tRNA, amino acyl tRNA is adding up. What is? Peptide bond is getting formed, peptide bond is getting formed between these two amino acids, and as you can see students, continuously there will be synthesis and formation of this polypeptide chain. I hope this much is clear to all of you. Next that you need to understand is how this process is going to end. So, as soon as the entire mRNA is translated, there will be a point where this translation process need to cease or need to stop, need to terminate. And again, in order to terminate, there will be requirement of termination factors, right. So, release factors or termination factors, what are these? These are actually proteins. Factors are nothing but proteins. So, release factor or termination factor will bind to the stop codons, right. Release factor when binds to the stop codon, that marks the end, that marks the termination or stop of translation process. And as a result, what will happen? The formed polypeptide chain will be released, but not to forget, releasing the polypeptide chain is not the end. That newly synthesized nascent polypeptide chain will now undergo into post-translational processing, where it will fold into its 3D structure, which is very important for this protein to become biologically active. So, newly synthesized peptide must fold into its proper 3-dimensional conformation to become biologically active, right my kids? So, after the formation, for example, this polypeptide chain is ready to be released. This is the polypeptide chain ready to be released. So, after termination, what will happen? This polypeptide chain will fold into a 3D structure, and the remaining subunits of ribosomes will be recycled so that they can be utilized again. So, that is the whole process of translation, students. I really hope now none of you will ever get confused on this concept of translation, right? If there is any doubt, do let me know. I would love to address your doubt. I would love to explain you any point you want me to explain it again. So, that is how the whole translation process works. Let me sum it out again for you. This is what? This is the initiator tRNA, you can say. So, this tRNA is going to be covalently bonded to this charged amino acid, hence there will be formation of an initiator amino acylated tRNA that will bind to the smaller subunit as well as that will bind to this mRNA. So, this mRNA binding to this smaller subunit of the ribosome and then binding to this initiator amino acyl tRNA. Finally, there will be formation of this complex where you have mRNA, amino acylated tRNA and smaller subunit. Finally, to this there will be bonding of larger subunit of the ribosome. So, finally, there will be a formation of a complex where you have large and small subunit of ribosome, mRNA and the initiator amino acyl tRNA. This is the initiation complex. This initiation complex, if you notice, there will be complementary pairing of this initiator amino acyl tRNA to the initiator or start codon, and then finally, in presence of certain initiation factors, with the help of ATP energy also the process of translation will start or initiate, and finally one after the other subsequent amino acid will join and there will be formation of a polypeptide chain. So, after the translation process you can see the protein get released and folds into a 3D structure, and remaining units of subunits of ribosome will be recycled. That is all, students, from today's session. I really hope you understood this. Thank you for joining this class, students. Do give me a thumbs up if you like me and this session, and do not forget to hit the bell icon so that you every time get notified whenever I will go live. Thank you, students. Bye bye. That is all from this class.